Yeah, thank you very much, Chris, by the invitation, by the opportunity to share our work with you guys. My name is Rubens. I work in the Department of Bacteriology in the lab of Jean-Michel Ané. Today, I'm going to talk with you about our projects involved biological nitrogen fixation in corn. I I split my presentation in three parts. Firstly, I'm going to give one introduction to the biological nitrogen fixation issue. Then I'm going to talk about our scientific approach, that is the genetic regulation involved in the formation of our roots. And then finally, our technological approach, that is our results involving our maize breeding program for biological nitrogen fixation. So let's start talking about the nitrogen. As you may know, nitrogen is a significant cost for farmers that grow corn. It's about 10 to 12% of the costs. These costs and the rates of nitrogen depends of several things like soil management and soil morphogenesis. It is also important, as Lindsay mentioned, like the rotation that the crops that we plant before corn. The level of technology of the farmers is also very important. And here I'm not talking about only about machines, but mainly about seeds. As you know, great hybrids, they request more a better fertilization. And also, it's important the yield that the farmer aims to produce. If we are making a simple math, let's say that we are expecting something about 200 bushel acres. That means that we need like 185, 190 endpoints and multiply by the price of the point of N. That's about 6 cents. And the area that we have in USA with corn, that's about 95 millions of acres, we see that we spend in USA something about 10 to 11 billions of dollars every year with nitrogen. Uh, but also the economical problem is not the only problem. As we saw in the presentation of Litze, there are a lot of problems involving the nitrogen required in the environment, like the acidification of habitats. One good example is the eutrophication in the Mississippi River Delta. Uh, and also we have like problems involve the health, especially the production of particulate matter that cause like significant cardiovascular and respiratory disease. If we, some colleagues from the University of Minnesota, they published a work a couple years ago that they estimated that these costs involving ammonia in like health and environmental costs, it's about 21 billions of dollars. So it's a lot of money that we spend uh, in this issue. So how could we use the environment in our favor? Probably you guys have seen this graph several times in your life. So we are like in deep in an ocean of nitrogen, right? About 78% of the atmosphere it's made of nitrogen. But unfortunately, the plant cannot access this nitrogen from the air. The plants can only access the nitrogen that we are put in the fertilizer. So, what kind of strategies could you adopt to make corn fix nitrogen from the air? Basically, we could like transferring regulatory pathways involved in nodulation from legumes. Here's a picture from Medicago. That's one legume that produces nodules. So we can like teach the plant the corn plant to produce nodules. But it involves like a very hard genetic engineer. There are a couple scientists work in that issue, but it's not something that we're going to achieve in a couple of years. Probably we will spend decades to have any success in this approach. We also could try to introducing the nitrogenase enzyme uh, from bacteria into corn cells. But again, it's another hard strategy that demands a big genetic engineer process. We could also engineer the bacteria, the diazotrophs that they are associated with corn. Just to mention, diazotroph is the name of bacteria that are able to fix nitrogen from the air. So we could engineer then to make them more efficient, but also it's a genetic engineer process. It would be faster than any genetic engineer process in plants, but we know that there are a lot of regulations involving the adoption of this kind of bacteria, engineered bacteria in the field. So the approach that seems to be most doable for us at that point is to explore corn varieties that they produce this mucilage, this gel that I'm going to explain to you how it's useful for biological nitrogen fixation. We use some corn from the state of Oaxaca in Mexico. That state, this part of Mexico, it's a very particular environment. It's like a high altitude place. Uh, it's a mountain. There are several villages in the mountain. They have a lot of fog, so it's super humid, the ambient there. And there are they have like people that are, it's a very rich in terms of culture. They use this kind of corn you see here that they produce a lot of arrowroots, and they secrete this kind of mucilage. And they use this mucilage for rituals and several things. But for us, our main interest is because in this mucilage, in the bacteria that lives here, we can take that nitrogen from the air, transform into ammonium, and then the plant can use this nitrogen. So how does the nitrogen fixation process occur? Okay, here we have the plant with these arrowroots and secreting mucilage. Let's imagine that this green illustration is the arrowroot that secrete the mucilage. In the mucilage, we will have like dissolved the dinitrogen gas, that is the nitrogen from the air, the atmospheric nitrogen. Then after here, it's where the bacteria acts, like the nitrogenase, the enzyme from diazotroph, they breaks this nitrogen gas into ammonia molecules. Like this is a very hard reaction. It requires a lot of energy, and just this kind of bacteria, they can do that. Also that we have protonation, so ammonia becomes ammonium, and then it comes inside to the corn tissues through the ammonium transporters. Here in blue, you can follow the nitrogen atoms. So after that, this nitrogen can go into the glutamine synthetase and glutamate synthase pathway. So here you can see the blue balls, the nitrogen. Here the nitrogen is fixed and can be used for corn for diverse metabolic activities. So that's how it works. But let's think a little bit deeper. If our goal at the end of the day is have high yield and low cost corn, we know that it's a function of three things: environment, plant, and bacteria. We know that it's very hard, to not say impossible, to modify our environment. So we need to work in plant and bacteria sides. Here we have in Jean-Michel Ané Lab, we have a big team of scientists that work. Some of the scientists work more in the bacterial side, and others, like me, work more in the plant side, and we have other scientists that work in the interaction between there. From my side, I'm more interested in corn, so I wanna discover how it happens. So we have two basic questions here. Why do some corn cultivars produce arrowroots and others don't? And how is mucilage produced and excreted? To the second question, my colleague Rafael Venado, that's now in CIMMYT, he has published some good results about that. his paper is in press, and so today I'm going to focus more in the production, in the formation of the arrowroots. Okay, now I'm going to the second part of the presentation, and we can talk about the genetic regulation involved in the formation of these arrowroots. To study that we started with like some double haploid populations. For those that are not used about that, we firstly requested seeds from these Oaxacan land races from CIMMYT and USDA, and then we crossed with a Midwest adapted inbred line. And then we have the F1 hybrid, and then we back crossed with the Midwest adapted inbred line to make easier the evaluation in field, because these Oaxacan land races, they hate West. Like they don't like when we plant them in corn belt. They have a totally different phenological cycle. Okay, after that, we crossed the BC1 hybrid with the haploid inducer line, and then select the haploid seeds, grow the haploid seedling, made the colchicine treatment, and then self-pollination, the homozygous D8 line, and then we have a population of D8 lines. We did that in partnership with Limagran, that's the French company. We did it 8 times, so we have 8 different population of D8 lines. So Limagran also helped us in doing the genotyping of these plants. What means genotyping? At that point, I would like you guys think, for those that are not used with this kind of technology, think about the genome, the DNA of a plant, like an address. Like we can find each gene, each region of the genome of the plant has its own address. So, okay, here it's the genotyping activity. And then we need
It's going to be over in about 6 minutes.
Yeah, yeah, then we can, okay, and then after making the genotyping, we need to do the phenotyping of these plants, of these 8 different populations. We have done it in West Madison, that's our field this year. We have done it also in 2023 and 2024. We assess several arrowroot related traits like arrowroot diameter, the number of nodes with arrowroots, that's one of the most important, the stalk diameter, flag leaf height, and flowering time. Flowering time, flag leaf height, and stalk diameter we use as a cover rates. In 2025, here in West Madison, we have like about 3,000 plots. We evaluated like 6 plants per plot. I mean, all these traits. So we have collected about more than 100,000 data. We are going to finish it hopefully next week. And we have a big team of about 20 students that helped us a lot. Okay, after crossing this genotyping data with the phenotyping data, we have a graph like this. Do you remember that I talked about the DNA, thinking about the DNA as a map? Okay, here we have the 10 chromosomes of corn, right? And so each region here has like different genes. So here, how to interpret a graphic like that? Here we have the logarithm of score, that it's the parameter that we use to make the quantitative trait loci mapping. It means that if we have some peaks like this, it says that in this interval here, we have genes here, they are likely to be involved in this trait. In this case of trait, they are the number of nodes with arrowroots. So if we want to discover the genes that are involved in this trait, that's the first step. But we can increase the search for this kind of genes, the important genes for our root formation. We can use it like put data from different populations. So in blue, it's from one population. In red, we have data from another population. We can add one more. We love when we find something like this, like overlap between two populations. It means that the probability of some gene here is involved in this trait, in the number of nodes with our roots, it's super high. But we also can improve this mapping, I mean, the search for the key genes. We adopt modern algorithms. Right now we are using one algorithm called the inclusive composite interval mapping. We use covariates to reduce residual variation, especially days to anthesis, flowering time. It's a super important covariate for us that help us to have better results. We evaluate like multi-parent populations, like nested associate mapping populations. That's a different approach. We compare these results with no gene annotation, so we use a lot the data in the maize GDB. We look for information on related species like sugarcane or sorghum, especially. And right now, we are not right now, we are going to start pretty soon the genome assembling of one of these land ratios. Also to try to improve to look for gene variations. Yeah, that's we also have another fine tuning mapping approach, but we can talk about it later. But now in the third section, I want to talk about the breeding program, our breeding program to introduce this trait, I mean the arrowroot related traits, into Midwest adapted inbred lines. So how did our breeding started? We started again, ordering seeds from CIMMYT and USDA, the Oaxaca land races, and we crossed the two Midwest adapted inbred lines. So we have the F1 again, self-pollinated, and then we have F2, and then we conduct it with several generations of self-cross. Then we have like some reels, recombinated inbred lines. We also conduct the breeding program through to back crosses, trying to like increase the adaptability of the plant to the environment in the corn belt. So we have different populations and recombinant inbred lines that came from the crosses between Midwest adapted inbred line and Oaxaca ladder races. So in that case, in the breeding, we evaluated all these arrowroot related traits, but also we evaluate agronomic traits because we don't want to plant that are propense to lodging, or they have like small ear size, or they are like resistant to some kind of disease. So we have much more things to evaluate in the breeding program. At the end of the day, we want to plant one inbred line with 6 to 9 nodes. 9 will be a dream, 6 is much achievable, forming these arrowroots and secreting this mucilage and also exhibiting good agronomic traits. We are at that point now we are splitting our breeding programs to have plants with different flowering times that we hope it can be used in different latitudes in US. We are dealing about with one polygenic trait, right? With moderate heritability. So it's a kind of hard to make breeding in that condition. So we need at different points of the breeding program, we need to make top crosses to help to increase the efficiency of selection. And our yield trials are conducted in Hancock. There we have like more than 100 hybrid. We evaluate that in 2 environments with standard nitrogen and with like about 40% off, like very low nitrogen. And also we have irrigation each 3 days because we need like moisture to get better results. And then we assess all these root related traits, chlorophyll content, and yield. One important step of our experiment in Hancock is to inoculate the diazotroph. As I mentioned, we have like a lot of students that help us to make the inoculation. Of course, in the future, we hope that in this technology, the bacteria could be inoculated in the seed. But here, for experimental purpose, we are spraying the bacteria in the plants to make sure that the bacteria is there. So we didn't harvest yet our field in Hancock this year, but we have like very nice results last year. We compared hybrids with similar backgrounds, sorry, but one hybrid made with an inbred line that has the arrowroot related trait and another hybrid that doesn't have this introgress trait. So and these results are about in the very low nitrogen place. I mean, like a lack of 80 points of nitrogen per acre. So here you have that you see that we don't have any good change in terms of chlorophyll content, but for some hybrids that has like more arrowroots, we see that the chlorophyll content increases very much. And you know that the chlorophyll content is related with the nitrogen status of the plant. But let's see the result in terms of yields, that we know at the end of the day, yield is the most important trait for farmers. Again, we compared hybrids with similar backgrounds, but the difference that one of the parent has the trait that's blue and another one doesn't have. So here we don't see too much difference, but when we go to our best hybrids, you see that there are a big increase in the yield under environment with very low nitrogen. And we assume that this increase is due to the bacterial association, this symbiotic association. When we plot the data about the number of nodes with arrowroots with the yield, we see a very super high correlation. What tells us that yes, when the plant has more nodes, and of course they have the bacteria inoculated, it tends to have like a bigger yield. To make sure about our data, we need to validate that in laboratory. So we make some experiments involve the bacteria. Here we have the different bacteria, and here the level of acetylene reduction activity. Basically, by accessing the level of conversion of acetylene to ethylene, we can quantify the activity of nitrogenase enzyme from the bacteria. So these three bacteria, Klebsiella michiganensis, Klebsiella varicola, and Azotobacter vinelandi, are the bacteria that we spray in the field. Here we have the controls, as you can see. They don't fix nitrogen. But also we make another experiment in greenhouses that we put a type of nitrogen, a fertilizer called the 15N nitrogen, 15N fertilizer. That means that this fertilizer has a different nitrogen. It's a label isotope. So you can track this nitrogen in the plant. What means that you can also, after you take the tissues of the plants and ground this, mill this, you can see how the percentage of the nitrogen that comes from fertilizer and then the percentage of nitrogen that comes from the air. So here the results are telling us that we have about 50% of the nitrogen that the plant used, it's from the air, which is great. In Oaxaca, in Mexico, these plants fix about 82%. Here in greenhouse, we got like 50%. We'll be super happy in the field in the future if farmers can reach something between 25 and 30%. That means something about $3 billion saved in fertilizers every year. So at the end of the day, we wanna like give another option for the farmers to save money and protect the environment at the same time. So I would like to talk my team, especially our PI, Jean-Michel Ané, our co-PIs. We have a partner in Georgia. We do the same experiments in Georgia to have two locations, and especially all the undergrads from our corn team and the workers from the agricultural research station, especially Janet and her team in West Madison and Paul and his team in Hancock. Uh, yeah, that's I would like to highlight that our project is funded by USDA, and I would be happy to reply and answer any doubt that you guys have. Thank you very much, Chris.
Yeah, thank you, Rubens. One question I had listening to your presentation was you definitely noted that the nitrifying corn in those low nitrogen systems, the yield got higher. So where is growing natively in Mexico? Are they um, following like the same nitrogen practices like in the US where we really put a lot of nitrogen on? Or is this better in a scenario where maybe it's difficult to fertilize or you have restrictions and you can't fertilize? Is this going to be a better corn to grow in the future?
Yeah, that's the goal. Use that, especially in like let's say poor fields where we don't like. I mean, for farmers that don't have condition to put like a super high inputs. We are starting to have some partnerships in Africa and India. We bet that it will be very useful in this place. But yeah, talking about Oaxaca, the big difference in Oaxaca is, well, this plant has a huge like phenological cycle that takes forever to flower. So they have more time to produce these arrowroots. And also they are very sensitive. I didn't have time to include more data, but we have some interesting data that plants in the edge of our plots, they produce more, they have more roots, and there's a big influence of the environment. We realize that it's not about the nitrogen availability in the soil and that it's not the moisture of the lower part. So our big bet now it's that could be light, could be a trick. And in Oaxaca, they don't plant like in USA in several rows. They are individually plants. So each plant, it's a edge. So for sure, it has a role in the straight. Yeah, so we are trying to figure out this like light influence. And yeah, there are like too much things to consider. in
I was
that.
going to say one question leads to another
Yeah,
question.
yeah, that's
So
the point. That's the
love it. Well, thank you for bringing an update to our crowd and our webinar attendees.